Hi, it's Maggie here again to explain to you the assay we use to quantify starch in a sample. Starch is often added to foods to play a role in texture. Starch is found naturally in foods too. See the starch in the ingredient list of this yogurt? And when you bite into an apple, sometimes it's floury, not crisp. In fruit, the starch breaks down to sugars during ripening, but early picking and cold storage can influence that ripening and starch breakdown. You will use a total starch kit to measure the amount of starch in your foods. The kit uses enzymes to break the starch down to glucose, and then uses a reagent to bind the glucose so we can measure the glucose. As we all know, a starch molecule is a chain of glucose molecules. First, we incubate our sample with alpha amylase, which randomly cuts the starch chains to make smaller molecules, which are called maltodextrins. We then incubate the maltodextrins with a second enzyme, amyloglucosidase, which breaks the maltodextrin down to glucose. Then we can label the aldehyde group on the straight chain of glucose with a tag called PABA, which absorbs in the visible region, so we use a spectrophotometer. Now, let's try it out. First, we weigh 2 lots of 0.1 gram of each sample into test tubes. Tap the tube on the bench to make sure all the powder is at the bottom. Next, add 0.2 ml of ethanol to wet the sample. Then vortex. Add 3 ml of the alpha amylase, which has been prepared for you. Incubate this in a boiling water bath for 6 minutes, stirring every 2 minutes. Then move the tubes to a water bath set at 50 degrees Celsius. Add sodium acetate buffer, 4 ml, then add the second enzyme, 0.1 ml of the amyloglucosidase, which breaks the smaller molecules into glucose. Vortex this and incubate at 50 degrees Celsius for 30 minutes. Afterwards, transfer the contents of each tube to a 100 ml volumetric flask. Use a wash bottle to make sure everything is transferred. Then make to 100 ml with water and mix very well. Take out 2 ml and centrifuge at 3000 rpm for 10 minutes. We will measure the amount of starch by measuring the glucose that is in this supernatant. While the sample's in the centrifuge, we need to make the standard. Make 1 ml of glucose standard at a concentration of 1 milligram of glucose in that 1 ml. Then pipette 0.1 ml into a centrifuge tube. The standard will be treated like the samples in the next step. Now we are going to measure the amount of glucose in the sample. For the assay, take 2 aliquots of 0.1 ml from each supernatant. Add 3 ml GoPod reagent to each tube and to the standard, and incubate at 50 degrees Celsius for 20 minutes. Then read absorbance at 510 nanometers. The calculation is quite a long formula, so please make sure that you have the absorbance from the sample and the standard in the right place. The final component of the equation is a correction factor for glucose as found in solution and in the starch. The difference of 18 is a water molecule. Good luck and see you next week.